Yeah, thank you so much. All right, so this lecture focuses on dynamic analysis or dynamic functional connectivity. And so I know you guys have recently covered concepts in connectivity, so yeah, we essentially extend these to look at more of the temporal dynamics. And so this is going to be more of didactic lecture, so I'm going to go a little more through some of the types of techniques and methods and trade-offs. So I will, give an overview first, talk about some methods for analyzing the data in a dynamic fashion, and then go into some of the issues and challenges in interpretation, and then wrap up. So functional connectivity, what is our goal? So we are interested in investigating how different brain areas may be interacting as systems or networks. And so we'll need a way of trying to infer these networks based on fMRI. And so we do, as you know, is we use our time courses. We extract the time courses from different brain areas, and we try to map their statistical similarities using measures like pairwise correlation, like Pearson correlation or mutual information or other similarity metrics. And hope that will give us some insight into the functional organization of the brain. So as you've probably heard about quite a bit already, there's a lot of stability in the patterns that can be identified across different people or across many different conditions. And so if you take the default mode network, for example, you can identify a pattern that looks quite a lot like the default mode network, not only in people who are awake, but you can still find it when people are asleep, although there's some differences. You can find it in other species. You can find it in infants as well. So one reason that these are so there's so much interest in studying these networks is because it seems to be some hallmark of brain organization that we can consistently detect. But if you look more closely at the actual magnitudes of correlations and topology of these networks, you can also see that there is variability across different conditions. Of course, we already talked about arousal states, but to illustrate a few other examples, it's in one experiment they asked how much is connectivity altered by what a subject is thinking during their resting state scan. So you can have, we just put in resting state scan, you have someone in the scanner and you say maybe, you don't give them much instruction, right? You say just relax. Some people could be thinking about their favorite song. Some people could be worrying about their grants or etc. And so it's actually kind of important to consider whether these just hidden cognitive processes might be shaping the connectivity patterns that we're getting. And so in this paper I highlighted here, they did this experiment where they would tell people to specifically do some of these common things, like they had to imagine songs in their head during the scan, or they had to be recalling memories. And then they asked, if you take the region-to-region connectivity patterns, can you classify what someone was doing based on that connectivity pattern alone? And they could actually get fairly good accuracy, meaning that the patterns do change according to what people are thinking. This is also useful to us in the context of, in clinical applications. So one of the primary uses of resting state is to ask, are intrinsic networks altered in disease? Can this be a biomarker? And the fact that we have these variations across disease, of course, is very important for being able to do this research. So the focus of this lecture will be on connectivity changes in time. And so I just want to start with this illustration. So suppose you have a subject resting in the scanner with their eyes closed, and it's a long scan, so they're in there for 54 minutes, poor person. Then you could make a connectivity matrix with that whole stretch of data, right? I could take my time courses 54 minutes long from my different brain regions, and I could correlate them and create this matrix. But then you can ask what happens when you start to break that down. So I could chop this up into the first 30 minutes or the last 24 minutes and so forth, and even 15 minutes, which is like, it's a long scan actually. Most of our scans are not 15 minutes, but even in that interval, you can still see some differences in the connectivity. And so questions that this might raise are, what is this coming from? Is it mainly arousal state effects or cognitive effects, or is there noise? What is happening that drives this variability? So this is an interesting avenue to think about. And so, oops. Let's kind of think about why we might just want to study these dynamic measures of connectivity. So one motivation is that we know that many conscious and unconscious processes are evolving on timescales that are shorter than one scan. So as we've already seen, different internal states such as vigilance, these are continuously shifting, and so there might be some real neural state changes that are happening over our data acquisition and could be very interesting to study. One thing to keep in mind, of course, is that we are using bold fMRI. So what we can study in terms of dynamics is, of course, filtered through this hemodynamic response. And so we are more limited to studying changes that are on the order of, a number of seconds rather than milliseconds, as you might be able to do with MEG or EEG, these kinds of technologies. But it's also been reported that, there are a number of using simulations, some faster state changes can even manifest in fMRI slower bold signals. So another motivation is that temporal features of our data may actually be important biomarkers. And so if we only look at the average correlation between brain regions over a scan, maybe we're losing a lot of information that could be useful to unpack and say, are there features that we can extract from, say, like a series of connectivity matrices calculated over the scan that might be valuable for understanding a disease? Maybe it's the variability in connections that's the more interesting feature than the average correlation for a particular application. And also, if we're studying these state changes in our if our focus of our research is on different states, such as affective states, cognitive states, physiological arousal states, then that's another motivation, of course, to calculating connectivity measures on faster timescales. And one thing to keep in mind is that in terms of the methods, it's quite exploratory because we have these 4D time course data, right? Like 3D data over time is an fMRI scan. There are so many operations we can do on this data to try to find features of interest. And so I think there's a lot of literature has been to just take approaches from maybe that are being developed in computer science, engineering, and other, for other applications, or in a basic, technique development for signal analysis, and say, like, how can we kind of leverage these techniques to understand more about brain activity? So there's a lot of cool work going on. And so over the years, there's been a number of studies probing whether features that you extract based on the dynamics of regional or network interactions can provide important information about individuals. So I just kind of put up this set of studies here that in one way or another has shown that some dynamic feature extracted from our data may provide a more sensitive measure than this kind of time averaged measure of connectivity. So for example, this study actually showed that if you look at, this is an autoregressive model of connectivity dynamics, it could explain more variance in cognitive assessments compared to this like longer time average static connectivity. But that was only true actually for certain measures. So it seemed that the dynamic features explained more variance in measures like fluid intelligence, sustained attention, working memory, some of these kind of where you have somebody doing an assessment and they're kind of doing some active cognitive or attentional task. But they also looked at other traits that were like like they had subjects give them self-report measures of things like how lonely are you, how stressed are you, and interestingly, some of those measures had a stronger relationship with not the dynamic features, but with the more time averaged, maybe stable ones. And so I think that what this suggests is that the different time scales about dynamics may actually be relevant for capturing different aspects of brain function. So the idea behind this dynamic functional connectivity analysis is, just to kind of put that in a picture, instead of taking all the time points in our scan and using that to make a single network or connectivity matrix or any other way of representing connectivity, then you can essentially consider shorter intervals of time and understand how these dynamics may be playing out in time. And so, although I have a picture here that indicates network or functional connectivity, more broadly, this field of dynamic analysis extends beyond these kind of region-to-region correlational matrices, but can be these signal patterns, more instantaneous patterns, et cetera, and I'll show you some examples of that. So, yeah, let's go through some methods. And I'll start with sliding window analysis because this is one of the most conceptually straightforward ways to think about studying changes in something over time. So this is also the basis for a lot of different approaches you may read about in the literature. So the basic idea is that we can divide our scan into some kind of overlapping or non-overlapping windows. And then for each window, you can just calculate some measure of interest. Like I mentioned, it could be correlations, it could be an ICA, the decomposition, things like that, over these different intervals of time. And so if you do that, then to illustrate some of the matrices that may come out, this study took different intervals of time in resting state data and found that, yes, just as we saw in the other example, there can be quite a bit of difference in the properties of the data over these different time windows. I made this movie actually back when I was a grad student and exploring data, but I like to show it anyway because I don't know, maybe it just like reminds me of kind of the things of like, I was trying to think about in terms of what this all means. But if you take a seed in the posterior cingulate cortex and you make a movie that captures how that map is changing over 2-minute windows of data, then you can see quite a lot of difference, even in the same scan. Something that changes most, actually, is maybe the anti-correlations. So those positively correlated regions seem to remain stable over time. And so at the outset, if you're just looking at resting state data, again, we have some questions about what exactly that means, but you can then definitely see that there are a lot of dynamics in data. So it's easy to just take your data and run some analysis like this, but there are a number of trade-offs to be aware of, and some parameters that you would have to select as well. And so one question you might ask is, how long do I make windows when I'm going to look at networks over time? On the one hand, if they're too long, then we may be missing something that's happening at a faster scale. But on the other hand, how short can we really make them, given that the hemodynamic response takes like 20-some seconds to return back to baseline? So if we slice it up too much, then we can get some weird effects happening just because we are taking very short sections of data. And also, the fewer data points that we have, the more unstable estimate we have to work with within each window. So there can just be more variability by virtue of having kind of the noise of shorter windows. And so those are some of the trade-offs to kind of consider. I would say that, like, in the literature, people tend to use around maybe 30 seconds as a short side when you're looking at when you're extracting some kind of correlation between brain areas. But we'll also look at some methods that look at more instantaneous variations as well. Another challenge to think about is when we start to open up this time axis more, then we're also opening up a much bigger space of features to work with, right? So we don't just have one connectivity map or network per person. We now have like a series of these things, and we got to mine them to figure out what is the essential information. How can we summarize those into different measures to gain some insight? And so suppose you have this kind of hypothetical measure of dynamic connectivity over time. This could be from a sliding window analysis, or it could be from another way of parameterizing connectivity change. But we can see that there may be some features that we can pick up from this dynamic behavior. So maybe how much do regions vary in time? How quickly do changes happen? And so basically can kind of ask, in a lot of cases, if you're studying this in the context of some clinical application or cognitive question, then it's really helpful to have some hypothesis that you're trying to test. Maybe there is reason to expect that there's more variability in these connections, for example, and so maybe you want to use this as a measure of interest. And then, just like we can do for a sort of typical functional connectivity analysis, a non-windowed one, we can take these metrics and then do statistical analyses across conditions or brain regions to understand how these are differing. So one study actually just asked which connections in the brain tend to be the most or least variable. Kind of nice basic analysis to do when you're first you're first starting to consider what dynamic features are present. So it turns out that, as you might expect, regions that tend to be very consistently correlated as parts of functional networks, those don't have a lot of variability in time because they show up very consistently by definition. But other regions, they seem to be more variable in terms of whether they're part of the same network or not. And so you can actually characterize those kinds of regions that end up shifting their allegiance between different networks. I'm going to skip this for now. But another way that we can look at structure in the data goes back to one of the analyses I presented in the last lecture, which is to see if there are certain configurations of connectivity that are represented in the data often. So this was a study where they took their fMRI data, and you can break it up into these windowed connectivity matrices, and then you can put those into something like k-means clustering and look at the centroids of the clusters that are detected. And they called these states, and you can do some things with this. So once you've assigned every window to the cluster it belongs to, then within an individual subject's data, you can ask how much of state 3 was there, how much of state 7 was there. In patients, is one state represented more than in controls? What did that state look like? What did it correlate with? And so these are the kinds of experiments that people have been carrying out in this with this kind of direction. And so one thing they looked at in another study from this lab, which is Vince Calhoun's group, was to look at the proportion of time subject spends in a given state and whether that distinguishes between groups. So they had schizophrenia and healthy control subjects, and they found that they could identify states where they were occupied more by controls than by patients, and vice versa. So now I'll kind of go to a bit of a extension of this method, but here we're going to move from chunks of data, sliding windows, down to individual fMRI timeframes. And so our raw fMRI data we can watch as a movie, right? Actually, it's hard to see much unless you normalize the signals because everything just looks like a brain in the raw form. But if you normalize the signals and you kind of just play out your data in movie form, it's interesting. You can see these patterns that are playing out. And so just like you can take a series of connectivity matrices and cluster them and see what the centroids look like. You can also just do that with individual time frames as well. So I can unroll the frames of a movie, and I can take these as individual, each one is an individual multivariate pattern, and I can also enter those into some analysis to understand what kinds of states are present. And so this is a nice illustration, courtesy of your instructor, Dr. Chen. And in this paper, we actually looked at these coactivation patterns in a couple of contexts, which I'll show in a second. But the idea is that if you take these individual time frames, you can cluster them, and you can find, the clusters will represent TRs that have a similar spatial distribution of activity. And so here's 3 examples. And these focus on clusters where activity in the posterior cingulate cortex is high. So one thing I wanted to emphasize here in terms of what, more information you can get from this analysis than a static connectivity analysis is that if you were to just take the PCC signal, use it as a seed, and calculate on average what does correlate with that, okay, you get a set of regions. But in time, when the PCC has a high signal activity, activity level, there could be one array of regions in the brain that also has a high activity level, and maybe those are areas of the default mode network. But at certain points of time, you might actually see like the PCC has a high signal, but another set of regions also has a high signal. So you're going to temporally decompose this PCC's coactivation with other brain regions, and that's more information can get from this data. So just like in the correlations, windowed correlation studies, we can also characterize, like in a given scan, how prominent different patterns are, these coactivation patterns. And one thing that she found here is that in resting state, compared to when you have somebody doing a working memory task for, say, 10 minutes, there actually seem to be a greater diversity of patterns that are represented, and probably because it's a less constrained condition compared to in a working memory task. And so we can take this and ask, there's a framework by which we can ask similar kinds of questions about patients versus controls, different brain states, how many states are coactivation pattern states are needed to represent a data set, and whether that tells us more about the dynamics of brain function. So this is one application from study in, actually was looking at depressive symptom severity, and this was a large sample of participants, again, with neuroimaging data as well as clinical measures. And a coactivation, the authors applied coactivation patterns analysis and found that the more time a person spent in a pattern where their activity was dominated by the default mode network relative to other externally oriented networks like a dorsal attention network, then it seemed that the worse their scores were in terms of depressive symptoms. And so, this one illustration of how you can maybe apply these in clinical context as potential biomarkers. So one thing that the CAPS analysis kind of ignores is temporal ordering. So if we are just taking our individual time frames and we're putting them into a clustering analysis, it doesn't matter which one was where necessarily, unless we start to look at that specifically. But we can actually start to look at whether or not there are repeating temporal sequences of activity in addition. And so this is work from Shella Kyles' lab, who's really done a lot in developing this area. And this is one of their early patterns or papers where they're proposing a pattern finding method for looking for these spatiotemporal patterns, and can find interesting, like basically little motifs in the data where you have maybe the activity of certain networks preceding the activation or deactivation of others. And so again, this might, if we find these kinds of repeating patterns in data, then that might indicate, oh, this could be something important that we can study, or something we might think could be altered in a disease state. So zooming from 2009 to 2021, this from the same lab, they extended this work to look at large publicly available data set, Human Connectome Project data, and could characterize the the spatiotemporal patterns that seem to be prevalent across individuals. And so I'm not going to really go into what all these mean, but one thing, in addition to looking at the patterns on the cortex, you can also see what is going on in deeper brain regions at the same time, subcortical regions, cerebellum. And so this is a dynamic signature. It's represented as something static here, but there is a propagating pattern across these brain areas. And so in some of the patterns, they saw some brainstem activation, followed by some cortical activation. And so it's a really powerful analytic technique for looking at these kinds of propagating waves. So in this slide, actually, I'm just presenting a collection of various methods that have also been applied in the field. There are quite a few different approaches that have been developed at this time. But one additional class of methods I could mention is change point analysis. So in the sliding window framework, you fix your window size and you just move that along. But why don't we let the data decide when some change happens? So there are methods from statistics where you can identify points in time where the statistics of the data are changing. Maybe that's of the raw signals or of the functional connectivity, but these groups have started to apply this to fMRI data and understand if we can detect these change points in a data-driven fashion. Another set of approaches that people are applying more and more are based on hidden Markov models. So these are designed to look for hidden states in the data and transitions between different states. And this was, some of the early work applying it to fMRI data. Their group also did a lot on, this is MEG data. So this is MEG data they have extended to fMRI since then, but can also characterize sequences of states and ask whether these can be altered in certain conditions. You probably talked a little about spatial ICA maybe. Temporal ICA is a variant that people have proposed for looking at other aspects of the dynamics. So instead of finding patterns that are statistically independent in space, we can also try to find temporal modes that are statistically independent from each other and understand what those look like. And for the complex network type approaches, this can also be extended into the time domain. You can actually have links between different time windows, the networks that you find over these different windows, and build multi-layer networks. So this is another rich area that's going on. So there's more too. There's no way to survey them all in a lecture, but yeah. So feel free to interrupt me with any questions as well. But otherwise, I'll just go on to this next part, where we're going to talk a little more deeply about challenges in the interpretation and also in some of the analysis aspects. So one question is, what do these changes mean when we observe them in our data? So, and how do we know that they reflect neural activity in the first place? And one reason to be concerned with that for fMRI in particular is we know that there's neural influences on our signals. And if at any point in time you see that some neuronal thing, like some head motion, is dominating over the signals you're getting, the real neurally generated signals, then that is going to perturb the pattern that you're going to get in a window of time. And it becomes more severe as we go down to these smaller windows. Then we're really sensitive to what is happening in the data at that particular moment in time. And yeah, so also interesting to understand the sources behind these metrics, and knowing that there can be spurious variability, then another important direction that's arisen as people have started to do this kind of analysis is to understand how we can appropriately do statistical testing for these effects. So in terms of non-neuronal perturbations, there are a few things that can cause some kind of a change in our data that affects something like a correlation map. One of them is taking a deep breath. So in the extreme case, if you take a deep breath and you just hold it, you see these really large bold signal changes. You can get much larger bold signal changes from taking a deep breath than you can by any task. So if someone's not taking that many deep breaths, then maybe that doesn't matter. But if you happen to be looking at a window in time around when someone is holding their breath or taking a deep breath for any reason, then you can see a change in the connectivity of your data. I know that there may be more lectures going into how these effects manifest in the data, but in the case of a deep breath, then the kind of impulse response function that has in the fMRI is some initial increase followed by a more delayed negative dip across much of gray matter. And so thinking from the standpoint of connectivity calculations, any effect where we are engaging regions in common is going to inflate our correlations between those regions. And so that would be the effect of this kind of manipulation. So yeah, it's actually, it's a good idea whenever possible. I can just throw this in there because I think it's a good message, to record physiological signals in your scans so that you know what's going on too. So it won't be as mysterious if you see some change like that. Another thing is that you can, just wanted to highlight that you can get meaningful looking variations even in random noise. So these are real fMRI time courses that I extracted from 2 different brain regions. And then over here, if you take their sliding window correlation, you can kind of plot what the correlation is between those 2 signals over time. But we can also make up our own time series by taking white noise, and we can convolve it with an HRF so it looks a bit fMRI-like. And you can do the same windowed analysis, and you might get the same amount of variability. So I just want to highlight that the presence of variation itself is not automatically some interesting feature. And so if we want to make a claim about the amount of variation or something else like the states or the coactivation patterns, then it's good to generate some appropriate null model for whatever feature that you're interested in. And there's more discussed in papers like this. With sliding windows, you can also get some spurious connectivity effects that relate to the window size relative to the frequencies in your data. So if you imagine that your data are a nice sinusoid here, it's hard to tell because this is a little bit subtle here, but this has a little bit of a shift. If we just look at these 2 signals and we say, we want to say that their interactions are not really changing over time, right? They're just 2 sine waves with a phase shift. But if you start to roll a window over time and calculate just the Pearson correlation, then indeed you're not going to really see much variation, but there's kind of tiny wiggles because of the phase shifts coming in. But if we shrink our window size down and we repeat that analysis, then you can see larger fluctuations in the connectivity values, correlation values that you're going to get between these 2 signals, because when your windows are small, then the phase difference is going to count for more. And so what are the practical implications of that? Well, it's been suggested that if you don't want to have these kinds of spurious windowing effects, then we want to use window sizes that have some number of periods of the slowest frequency of our data in it, so that we don't have any kind of component that's slow and are giving rise to this non-stationary effect. And so it was actually recommended in this set of papers that you could high pass your fMRI data so that the window length, if you know you want to use a certain window length, like 30 seconds or 100 seconds, then you could actually filter your data so that it doesn't have a lot of the slower frequencies, so that you're sure that you're focusing on the ones that are meaningful in the context of the window size that you're looking at. Also, we want to ask, what timescales should we study for looking at connectivity changes? Of course, in terms of the analysis, one trade-off is, we'd like short windows, but that would give us fewer data points per measure. But also, bold signal doesn't give us a lot of guidance in terms of what time scale we should study, right? It's not like we see these oscillatory peaks in bold like we do in EEG algorithm, for example, where it's like, okay, we want to look at algorithms. In fMRI, data actually tend to have this 1/f type characteristic. And so we have contributions from a lot of different frequency components, and just from the data itself, it's not so obvious what time scale we want to focus on. And so I think the experimental question comes into play here a lot. Are you interested in studying something that's playing out on very long time scales, or is it something that you expect to happen much more quickly? So that can also guide what you're looking at. But in general, it's kind of hard to say just from the characteristics what frequency we should be focusing on. Another approach, I guess, is to take a more data-driven avenue and say, well, I may not know what scale I want to look at, but I can actually make a portrait of interactions between brain regions across multiple time scales at once. And this can be powerful visualization, especially when you're trying to understand, if you're the first person trying to apply some dynamic analysis to a specific context, you might want to get a sense of where is the variability happening in terms of the frequency ranges. And so you might explore that and see that, using something like a wavelet analysis, which decomposes, in this case, coherence as a function of both time and frequency, you may be able to see that the interesting changes are happening at certain particular frequency band. On the other hand, just because we're talking about trade-offs, while this can be a really rich representation of our data, there's a lot of information on these kinds of graphs. So not only do you have some magnitude, so firstly, not only do we have time and frequency instead of one value that says how correlated are these signals, but you also have across this plane maybe a phase difference between the signals. And whether or not you're interested in all that may really depends what you're looking at, but it does give you the option to look at all of these relationships. When we're studying connectivity, it's always important to think about what's going on in the raw signals themselves, because two signals correlated at 0.4 may actually have very different things going on in terms of their time signatures. And so, and if you have an amplitude change in the data, then and it suddenly becomes higher, much higher than the noise floor, that might also, govern the kind of correlations that you're seeing. So in some cases, you can look through correlation matrices. Sometimes it seems the signals are just not that much changing in amplitude, but they are becoming more correlated. Other times there's amplitude changes mixed in. So it's a lot to think about. This is complex, but I mean, it's just to keep this in mind when you're looking at connectivity values and thinking, there may be more to, it may be important to kind of unpack any connectivity value into thinking about what might be going on in a more raw form. And connectivity, so we kind of covered a range of different methods so far in the examples that I've shown, but there are so many different ways to represent data, and even though taking correlations between regions is really common, sometimes a representation that might be kind of truer to what's going on is something like an event or a traveling wave or a frequency shift, and so I think that's also worth considering. So for this last bit, I wanted to discuss a bit more on what are the sources of variation that are happening to drive connectivity changes. So supposing that we're looking at neural driven bold signals for now. Recording other measures during the scan can be really helpful in order to understand that. So as illustrated earlier, you can look at EEG, you could look at cardiac and respiratory signals, physiological signals. There's, especially in resting state, when you don't have a lot of active behavior, it's really hard to get a probe into what somebody's state is. So these recordings are just so helpful to have. I'm always happy when I've recorded them in a previous study because it always gives some useful information. I'm going to skip this because we did that one last time, but when you're going back to this topic of when you're maybe not able to record measures, people have been asking, how to what extent can you use dynamic connectivity or activity patterns to track someone's brain state? And this was one of the first studies that, at least that I'm aware of, to try this in resting state fMRI, but in the context of sleep. And so they did record EEG simultaneously with fMRI. So the EEG kind of provided this gold standard for what sleep stage a person was in. Were they awake? Were they in REM sleep, etc. And then they took sliding window connectivity patterns, and they asked if they could put this in a support vector machine classifier and match what they were getting based on the EEG-based sleep staging, and found that they could do that pretty well. So people have actually, in the course of this field, there have been questions asked about what these dynamic connectivity changes mean, and several papers have actually focused on the effects that we spent the last lecture on, which is arousal effects. But this one, just to show another illustration, was asking how much the questions or how much the windowed connectivity patterns relate to sleepiness. This is long scans with EEG, but they could find that, the global correlation patterns that appeared sometimes in the data were indeed linked with states like light sleep. It's also very interesting to investigate how dynamic fMRI patterns connect to other types of mental processes, and mind wandering is another one that is very prominent when people are in the scanner. But how do you figure out whether somebody's mind is wandering? I don't know. Does anyone have any ideas? Yeah. Yeah, well, that's one way to kind of indirectly probe it. Like if you have someone doing a task, like if they're kind of lapsing in their performance, that might be one contributor. It's kind of hard to tell, but this study actually kind of just presented some probe every once in a while. So they kind of introduced this additional task that asks, how much was your mind wandering just now? And the subject had to make a response. I think they kind of ignored those parts when they did the data analysis, but here they just kind of asked them at certain points during the scan, or you can give them a questionnaire to ask afterward. It's really hard to kind of probe these internal states, so without disrupting the system, right? So this is actually quite challenging to do research in. But I think this study, they looked at the amount by which they were daydreaming, and saw that this correlated with the amount of correlation between the parts of the default mode network. So we find this really interesting. And I focused this, a lot of this work on resting state data, but you can actually look at experimentally induced changes in connectivity or in any other dynamic metric as well. These are two examples, and in the first one, they actually had people watching sad movies in the scanner, and they were actually interested in affective responses. So they would isolate sad scenes in the movie and then look at limbic network connectivity. But we're also just interested in the dynamic behavior of the limbic system's connectivity over the course of the scan, and how that correlated with measures like heart rate variability. And in this one, this was a pain study, I believe. So they actually gave in an IRB approved way, pain to a participant over time, and tracking a brain response to pain is also a really important question. So they knew that when the stimulus would come on, they could kind of track the dynamics as people's sensation of pain maybe ramping up or going back down, and the network activity that goes with that. So this is the summary. So there's many possible methods that can be used to explore bold dynamics. Of course, keep in mind that we are looking at certain timescales. Very, very short windows, analysis windows can be sensitive to noise or other spurious variations. But there's increasing evidence that these descriptions of data based on dynamic features can provide useful biomarker information, information that complements static connectivities and not just recapitulating the same stuff. And we can design experiments where we study dynamics of network connectivity together with specific manipulations, or we can study them at rest. So some open questions I wanted to pose to you, in case you were doing research on this. So one thing that is consistently that people consistently grapple with is that now we have a lot of high dimensional features. So isolating the ones that are most relevant for a given scientific or clinical question is always important. There are also other ways of getting spurious coupling between fMRI time courses, even the absence of noise and artifacts. And so how to best isolate dynamics of functional relevance continues to be an ongoing question. And also, there are, it's not like brain, some of the models suggest that the brain is like in one state and then switches states and things like that. But it's pretty complex. There's probably a lot of continuous overlapping processes. There have been some models proposed that say at a given time point or window, there's a mixture of states, and we can represent this mixture of states. So there are a lot of overlapping processes that may be on different time scales, and so looking to some of the different analysis methods, not all of them will be equipped to kind of handle that. So I think there's still more work needed on how to identify and disentangle overlapping processes. So we shared the slides, I think, but there put some references here in case you want to see some of the review papers on this topic.
Is there anything you have to keep an account for if you're looking at, you're trying to target lower frequency dynamics? Like, for example, I work in a sleep lab, so we're interested in CSF and MRI, so slow processes. So how do you balance that?
Yeah, well, I mean, if you're interested in looking at, I guess first, even if you know that there's some change happening on a slower time scale, but the signal that you're looking at is not directly tracking it, you can still remove the slowest frequencies and focus on what else is going on, maybe in terms of the other frequencies during those times, if that makes sense. I don't know if that kind of answers your question, or it's like, but yeah, there are slow, naturally very slow processes, and then maybe getting longer and longer scans is helpful for that. But then, I guess depending on the timescales of what's happening across those changes, you might have some analyses that look at you might do multi-scale type analysis where you're looking at larger windows with slower variations, and then adjusting the window size to look at faster ones.
How much should we worry about data quality and things like CR when it comes to considering
Oh, yeah.
actually use?
That's so important, actually. Yeah, this is a great question. So the TR kind of, so data quality always best we can get is helpful, especially for these dynamic analyses. But for TR, there's been a lot of interesting discussions and trade-offs pointed out. Actually, Dr. Chen has written a nice article on, as we go to, so when these fast fMRI techniques start to emerge, like people think, oh, we can scan faster and faster. Of course, that has other impacts on the ability to measure certain other features. It may improve our ability to see some things, but at the same time compromise others, which I can't maybe summarize very well in a response here. But there are complex trade-offs in these. So thank you for raising that, because that's a really important point. Okay. Yeah, I mean, one thing is like, it would be great that the more time points we get, the better. But yeah, it's not quite as straightforward as that. So. Okay. She could answer that, though, in another lecture. Oh, yeah.